Hello students, today we are going to learn Principles of Inheritance and Variation Part 5. In this part, we are going to learn in detail about karyotyping and pedigree analysis. Now let's see what is karyotyping. It is a technique through which a complete set of chromosome is separated from a cell. They'll take a cell and the complete set of chromosomes are separated and they are arranged in pairs. That is called as karyotyping. The diagrammatic representation of the chromosome, we call it as ideogram. This is, if you see this picture, this is actually the human karyotype male and this is the human karyotype female. If you take the male cell and the complete set of chromosomes are separated from the male cell, and when they are arranged in pairs, this is how they look like, and the female chromosomes will look like this. And the diagrammatic representation of these chromosomes, we call them as ideogram. We call that as ideogram here. So if we take the human chromosomes, they are totally divided into 7 groups: A, B, C, D, E, F and G. These are the groups. If we take the group C, is the largest. It includes more number of chromosomes. At the same time, X chromosome belongs to group C and Y chromosome belongs to group G. So very important, children, remember this. They may ask which is the biggest group. Is a group C here, to which group the X chromosome belongs and to which group the Y chromosome belongs here. These are all important 1-mark questions, and this is what the karyotype is. Now let's see how the karyotyping is done. Let's learn preparation of karyotype. Jayo and Levan, in 1960, they described a simple method of culturing the human lymphocytes. Okay, blood in from human blood, they have isolated the lymphocytes and they have cultured the lymphocytes here. This simple method was identified by these two people. And after culturing the lymphocytes, mitosis is induced. At the metaphase stage, when the mitosis is at metaphase stage, if you take mitosis, we have different stages here, but particularly at the metaphase stage, the cell division is arrested. The cell division is stopped at the metaphase stage. How? By adding a chemical called colchicine. Colchicine will arrest the cell division. And at the metaphase stage, the colchicine is added to arrest the cell division. Why the cell division is arrested at the metaphase stage? Because the chromosomes at the metaphase stage are very clear and they are clearly visible here. And after that, a suitable spread of metaphase chromosomes is done. Here you can see a suitable spread of metaphase chromosomes is done, and then it is photographed. After that, the individual chromosomes are separated, are cut from the photograph. They are cut from the photograph and they are arranged in orderly fashion in homologous pairs. So after taking the picture of this, they will cut the chromosomes and they are arranged in orderly fashion in homologous pairs like this here. And you may ask how they will identify the homologous pairs here. This is done by chromosomal banding. So chromosomal banding will permit the structural definitions and differentiation of chromosomes. Based on chromosomal banding pattern, they will do this here. And this is how the preparation of karyotype is done. Let's learn the application of karyotyping. It helps in the detection of chromosomal aberration, that means change in the structure of chromosome, maybe because of addition, deletion, duplication or translocation here, and identify the loss and addition of chromosomes. So if a chromosome is added or it is deleted or lost, it can be identified by the karyotyping, and it helps to detect the aneuploidies. Aneuploidies means gain or loss of the entire chromosome here. So if you take in this condition Down syndrome, trisomy of 21st chromosome, homologous chromosomal pair happens here. So instead of one homologous pair, you can see 3 chromosomes here, and this is a aneuploid. Here one chromosome is gained extra here, and we call this condition as Down syndrome, and it is a genetic disorder, and it helps in the gender identification. It also helps in pre-birth diagnosis of genetic disease. So if there is any genetic disease, if a family is having a history of genetic disease, that genetic disease can be identified when the baby is developing in the uterus itself. It can be diagnosed in the mother's uterus itself whether that baby is having genetic disease or not. This is all about the applications of karyotype. Now let's learn the types of chromosomes. Depending upon the position of centromere, this is the centromere, and the relative length of two arms. The relative length of two arms, this is one arm and this is the second arm here. The relative length of two arms. The human chromosomes are of 4 types here: telocentric, acrocentric, submetacentric, and metacentric. Coming to telocentric, children, here they call them as P arm, and here they call it as Q arm here. In telocentric, the P arm is absent. There is no P arm, and the centromere is located on one end. One end the centromere is located, and there is no P arm. And coming to acrocentric, the P arm is very small, and the centromere is near the end. It is very near the end. You can see here the position of the centromere. And coming to submetacentric, the P arm is little smaller than the Q arm here, and the centromere is in the middle, almost in the middle here. And coming to metacentric, the both P and Q arms are equal in length, and centromere is exactly at the middle of the chromosome. Okay, if they ask in 1 mark question, if the centromere is at the extreme and what is the chromosome called as, like this type of questions you will get, children. And this is about the types of chromosomes. Let's learn about pedigree analysis. We're going to learn in detail about this topic in this session. Let's define pedigree. Pedigree tree here. Pedigree is a family tree drawn with standard genetic symbols. So there are certain standard genetic symbols. With that, this pedigree, the family tree, is drawn. And why it is drawn? To show the inheritance pathway of specific phenotypic character in a particular family here. So if you take, this is the pedigree chart here. So it is the family tree which is drawn with standard symbols here. These are all the symbols. I'll teach you about these symbols in detail in the next slide. And it shows the inheritance pathway of a specific phenotypic character in that family here. And pedigree analysis is the study of traits as they have appeared in a given family line for several past generations. So if you take, this is the family, okay, it's a pedigree chart here. It helps, pedigree analysis is the study of traits. Traits means character as they have appeared in a family line. So if you take, this is generation 1, this is the generation 2, this is 3 and 4 here. So in these 4 generations, how a particular trait is appeared in a given family line for the past generations can be studied by this pedigree analysis. These are the standard genetic symbols we were talking about in the previous slide. The male is represented as a square. If it's a square, then it is male. And for female, it is a circle. The symbol for female is circle. And the marriage or mating is represented like this, a line between a male and female. And the parents and children, if you take, this is mating or the marriage, and if the line is joined like this and it is shown drawn like this, then these are the children of these parents. Here the square, I told you, it represents it's a symbol for male, one boy child and one girl here. And next, suppose if this is father and this is the mother, and then the symbol is like this. The symbol is like this. Okay, not like this. The symbol is like this. They are twins, but so you can see in this case, in this case, in this case, they are all twins. Here one boy and one girl, here two boys, and here two girls here. They are the twins, but they are non-identical twins. The non-identical twins will be represented like this in this, okay? In this, a boy or girl, two girls, or boy and a girl here. And if the symbol is like this, so a join here, they are the twins. If an additional line is there like this, if an additional line is there, they are identical twins. We call them as identical twins or monozygotic twins. And if the symbol is like this, then the sex is unspecified for them. And if there is a number, if in the symbol square 2 is there, then two boy children, number of children here. And in the circle, if there are 3, then circle is a symbol to represent the female and 3 girl children here. And if the square is darkened or the circle is darkened, square is for boy and it is the affected male, and the darkened circle represents the affected female. And if they are half dark and half light, in this case here, they are heterozygotes for autosomal recessive character. They are heterozygotes for the autosomal recessive character. And in a circle, a dot is there. Then it is a carrier for sex-linked recessive disorder. It's a carrier for sex-linked recessive disorder. And if a cross is there, then it represents the death. It represents the death. And then if the symbol is like this, it represents the stillbirth or abortion. And if it is like this, you can see this is prospositus, an arrow with darkened square, or it can be an arrow with darkened circle. If there is an arrow, and again suppose if it is darkened alone, they are the affected individuals. If there is an arrow facing this darkened square or the circle, then it is called as prospositus. Okay, what is this prospositus? The initial subject presenting a mental or physical disorder who causes the study of his or her heredity in order to determine if other members of the family have had the same disease or carry it. So let's say in a family, suddenly a person is having a disease here, and he is the initial cause to study the family tree. So how the family tree, is there any disease in their grandparents or in the siblings or in the parents, or are they carriers? Actually, he is initially leading to the study. He is the cause for the study. He is the initial subjects for the study of the family tree here, and we call that as prospositus. And if you take two lines, okay, two lines, a square and the circle, male and female joined by two lines, it is consanguineous marriages. What is this consanguineous? So consanguineous means a marriage between close relatives. So you can see like marriage between the close relatives within the same family, and that kind of marriages we call them as consanguineous marriages. Okay, these are all the genetic symbols used in pedigree chart here. Now let's see how the pedigree chart look like. This is how the pedigree chart looks like. This is the generation 1, 2 and 3, 4 here. Now you have to answer the following questions here. Now, how many individuals, how many affected individuals are there in generation 3, and how many carrier females are there in generation 2, and how many normal males are there in generation 4? So find these answers and attend for the next class. Thank you, children.